Our final speaker before the break is Ed Clark. Ed was an undergraduate, or a graduate student, at the University of Pennsylvania in Joan Brugge's lab, where he started his work on Sark family kinases. He then came to the MIT Center for Cancer Research to study metastasis in Richard Hines' lab as a postdoctoral fellow. For the last 12 years, Ed has worked on cancer drug development in the biotech and pharmaceutical industry, first at Millennium Pharmaceuticals, then at Bristol Myers Squibb, and now at Merck Research Labs. He's currently the Executive Director of Oncology Discovery and Preclinical Sites Lead at Merck. Ed has played a lead role in the preclinical testing and clinical development of a number of important new anti-cancer therapies, including cetuximab and dasatinib, which is a new generation Sark inhibitor that's showing great clinical success. His talk today is entitled From Targets to Drugs for Cancer Patients. Welcome.
Great, thanks, Mike. It's great to be back here at MIT, and I want to thank the organizers for the opportunity to come back and share some memories with friends, colleagues, mentors, and people that sort of grew up with. I noticed in Richard's slide the other day that I still had facial hair and an earring, and those things are gone now. But what's not gone is the zeal, the excitement about great science, and we've seen a lot of that last couple days. It's something, while I was here at MIT, was something that was really fostered here. And one thing that's continues to be exciting to me is taking great science and then translating that into results in the clinic. And hopefully that excitement shows up here today in my presentation, because I think it's something that many of the people who work in the biotech and pharma industry are excited about. So what I want to talk about today is the important role that science can play in what goes on in the clinic. And I'm kind of breaking this talk up into three parts. One is on the past, where progress has been made over the last decade, the things that we've been able to do. I think it's always good to point to where there has been progress made, but also use it as a backdrop to show where additional progress needs to be made. The real focus of my talk today is the need for combinations of agents to be used together. You'll see this word combinations appear many times in my talk today, and that's really what the focus is. Despite the advances we've made with single agents, I think most people agree that it's about combination therapies that will really help patients. And that's what I'll give you one vignette of a combination of two agents that are showing great effectiveness now in patients. And then, while I don't have a crystal ball, I would like to provide at least one vision of the future about how best to do this type of work, to combine agents together and to provide hypotheses that can be tested in the clinic. So I'll start, I don't go back quite as far as some people do, but I remember, and many of you may also remember going back to some of the meetings in Frederick, Maryland. And at the 10th annual Oncogene meeting, I remember Mike Bishop giving a presentation and lamenting back in 1994 about, for the moment, we cannot assign distinct steps in tumorigenesis to individual genes. Well, lots happened since 1994. Eric Lander referred to it yesterday when he talked about some of the new technologies that in 1994, or even in the year 2000, were very nascent, are now standard technologies that are used by many labs around the world. So I'll show how we're using these various technologies to help in progress of developing therapeutics. I think one of the other things that's been really important and has changed a lot since 1994 is the training of clinician scientists. Nowadays, you go to meetings like the American Association for Cancer Research, and there's as many clinicians there as there are basic scientists. And I think that's been very important, and I think MIT has been in the forefront of training many of those clinician scientists, several of whom have presented here today and over the last 2 days. One of the other things that's progressed significantly is a systemic understanding of molecular mechanisms of tumorigenesis and therapeutic resistance. We now understand the role that some of these genes like the epidermal growth factor receptor play in sensitivity in lung cancer, about RAS and its role in resistance in colorectal cancer, obviously about BCR-ABL and the important role that it plays in CML, and more recently, genes like RAF and the mutations and the role that they play in various diseases and agents that are being developed against the cancers, particular melanoma. So I think all of this has led to an explosion of new therapies that are currently in testing. And one estimate is there are probably right now about 800 agents in clinical development for cancer. And I just had to throw in one slide there to show that these new technologies were adopted way back when Richard and I were in the lab. But in terms of the types of agents that have been developed over the last 10 years, here's a list of 33 agents that have been developed for cancer patients. And I want to highlight some of them down here below, which have, I think, really made a difference in patients' lives. So in CML, if you had the disease back in the 1990s, your 5-year survival rate was about 40%. With the introduction of first Gleevec and then 2 agents, Sprycel and Tasigna, that 5-year survival rate is greater than 90%. And it's led some to speculate, such as Charles Sawyers, who has led the development of several of these compounds, that you're probably just as likely to die being hit by a bus as you are of dying of this disease if it's caught early. Colorectal cancer is another disease which back in the 90s had about a 9% 5-year survival rate. Now that's more than tripled. And that's in part due to the development of new agents to treat those patients. But I think everyone who works in this area would concede that for many advanced cancers, much more effective therapies are needed for these patients. So what are the challenges in the development of effective therapies for patients? Well, as I've already pointed out, most of these agents have very modest effect as single agents. Obvious exceptions are in cases like in CML, when it seems to be a single gene drives that disease. But even in more advanced disease in CML, in the blast stage, where many more genes are involved in tumorigenesis, that it's a single gene blocking that is not sufficient to really treat the disease. And this is largely due in part to drug resistance, probably due to functional redundancy and pathway crosstalk that causes patients' tumors to be resistant to tumorigenesis and to the treatments that we provide for them. Now, most treatments have demonstrated clinical benefit in the setting of combination therapy, and I think, again, you're seeing the word combination there again. But typically, the way things have been developed is to add a given agent on top of standard of care. So some combination of drugs works, we add one more drug on top of that and hope to get some incremental benefit. Now, this is based in large part on the ability to get drugs registered and the easiest path forward, but it may not be the most scientifically desirable way to move forward. And I believe that recent understandings of the mechanisms underlying malignant transformation, the sort of scientific principles that you've seen in the various talks given over the last several days, provide an opportunity to design novel combinations of targeted therapies to treat these cancer patients. So here's just one simple example of signaling downstream of a growth factor receptor. If in this case you block a single node in this pathway, say the AKT pathway, by blocking with an agent that blocks it, what you see over here on the right is that you can block AKT signaling just fine. But what you see is that other pathways are not affected as significantly. Whereas if you block multiple nodes, in this case we're blocking both the AKT pathway downstream of growth factor signaling, as well as the MEK pathway, MEK-MAP kinase pathway, that you can see greater inhibition of cell signaling. So for instance, if you look at phospho-S6, here you see much greater inhibition in the combination of an AKT inhibitor plus a MEK inhibitor. So this is the sort of thing that we'd like to be testing both preclinically and then eventually in the clinic. But how do you figure out what combinations to use? This slide highlights the problem that we have about combinations. And it's a lung cancer example using 2 agents that are now standard of care for lung cancer, erlotinib and bevacizumab. And if you just had, say, 5 drugs in your pipeline that you're looking to combine with those standard of care agents, That provides you 14 possible clinical trials to be run if you were to combine those agents together. Now, if you also decide, well, we don't want to just treat everybody with lung cancer, we want to identify specific subpopulations and pick 4 somewhat well-known markers in lung cancer, that provides you another 16 possible patient subgroups if you look at combinations of those different biomarkers. So that, if you add that up, could provide a total of 224 possible phase 2 clinical trials. So again, the problem statement here defines how do we define the optimal combination of agents to be used in specific disease context. Now here shows one example of what could be done if you had enough money. This is an actual phase 2 trial of a company developing an IGF-1R inhibitor, which shows a total of 12 different arms in this phase 2 trial, each with 20 patients in it, each combining with different targeted or chemotherapeutic agents, and hoping to find some activity somewhere. Probably not the best way to go forward. What you'd like to do instead is perhaps generate a hypothesis based on preclinical experiments and then test that in the clinic. And so one way to identify rational combinations is to use some of those new technologies that have been identified over the last decade or so, including RNAi-based libraries, mRNA profiling, copy number, DNA sequencing, much of which has been talked about over the last 2 days in talks here. And today I'll give an example of how we're using these different technologies and high-throughput robotics to look at one example of a combination we've identified now preclinically, tested preclinically, and now moved into the clinic with results. And this is a particular combination of an mTOR inhibitor with an IGF-1R inhibitor. And what I want to show you today is how the combination was first identified, confirmed, and then how we tested it preclinically. I'll show you clinical results that have just recently been presented, and then talk a little bit about what the paradigm for future combinations, how you might go about doing the discovery and development of combinations. So I'm a little intimidated to show this slide here, knowing that David Sabatini is somewhere probably in the audience. But for many of you, you know the mTOR pathway and this TORC complex here that's blocked by rapamycin or rapalogs. The particular one I'll be talking about today is rhodafrolimus, which is a rapalog that we have now currently in phase 3 development in sarcomas, but looking to better understand where else to develop this agent. And so what we did to identify potential partners for mTOR, rhodafrolimus, is to take cell lines treated with the agent or untreated cell lines and use the same sorts of synthetic lethal approaches that have been shown several times here, but to do it in this sort of enhancer screen library, where we're looking for agents or siRNAs that when in combination with mTOR will actually show greater inhibition of cell growth or of signaling pathways than when you just use the siRNA alone. And identify candidate enhancers that then will be further validated in subsequent studies. So this is the approach that we took with our mTOR inhibitor. And what we identified first is when we looked through a screen of siR-based libraries, that the number one hit in the screen was insulin-like growth factor receptor 1. Now, this was exciting to find a combination. Also was exciting because we happen to have an agent in clinical development against IGF-1R. But what was even more exciting was the fact that when we did the reverse screen, when we looked for hits in a library against that would enhance IGF-1R inhibitors, what we found is the top hit was mTOR itself. So in 2 screens looking for enhancers of agents, we found that they both showed that in the IGF-1R screen, you found mTOR, and the mTOR screen, you found IGF-1R. Now, I think people who have done these sorts of screens recognize that most of the time you don't get this sort of synergy. So this was pretty exciting to us. The next step was to show in vitro that this hypothesis held up, not just in the screen, but in a panel of different cell lines. And so what I'm showing here is data for 3 different cell lines, which, as you can see, express different levels of the insulin-like growth factor 1 receptor. And what was noted was that when you treat with an mTOR inhibitor, that, as expected, you can block very nicely phospho-S6 kinase downstream of mTOR. And But what you also noted was that there seemed to be a feedback mechanism when you treated with an mTOR inhibitor, that you actually saw induction of AKT, phospho-AKT. So there's a feedback loop that seems to be occurring when you treat with a rapalog that inhibits the TORC1 complex. But what was noted here was that when we added the IGF-1R inhibitor to treatment with the mTOR inhibitor, that you saw inhibition of that signal, that phospho-AKT signal, as well as the phospho-S6 kinase, suggesting that you're able to inhibit both of those signaling pathways much more than with either agent alone. Now, what we like to say often is that if you see an effect in one cell line, that's like seeing an effect in one patient, and if you have an effect in one patient, you don't have a drug. What you want to see this is in large panels of cell lines, and also to give you a better sense of which patient populations you might want to be treating. So what I'm showing you here is data on the combination in a panel of 67 different breast cancer cell lines. And so the cell lines are shown here, obviously much too small for you to read. But what you can look at is the gene expression profiling, and in particular of the estrogen receptor, which, as most people know, in breast cancer really drives the expression of many genes. And you can see these they tend to block into those that are ER positive and those that are ER negative. And what I'm showing here in these little black bars are the ability of either mTOR inhibitor alone, IGF-1R inhibitor alone, or the combination of the mTOR inhibitor plus the IGF-1R inhibitor to inhibit the cell growth. And so what you'll see is if you have a large black bar like this one here, it suggests that the agents in combination are doing absolutely nothing. It shows that if you see absolutely nothing there, it suggests that in this case, for instance, the IGF-1R inhibitor alone is completely blocking the growth of that particular cell line. And what was noted was when we looked at the combination of these two agents in the 67 breast cancer cell lines, that you saw the most activity in this set of ER-positive cell lines. Not only those that were ER-positive, but those that also had a high proliferation index, the so-called luminal B subset of breast cancer. So the next thing we wanted to do is take this in vitro results in vivo, and here shows results for just one model. This happens to be a lung cancer xenograft that was tested, and you can see when we use the mTOR inhibitor in red here, the IGF-1R inhibitor in blue, you see some activity relative to the untreated control, but you see the combination of the two actually shows regressions, and rather than just slowing of the growth of those tumors. And it does so in a dose-dependent manner as shown over here. So when we add greater amounts of the IGF-1R inhibitor, first of all at 0.2 mg per kg, 2 or 20 mg per kg, you see really frank regressions, again, in a dose-dependent manner. So we have both in vitro evidence and in vivo evidence that the combination could be active, particularly in a subset of breast cancer, but also, as I showed you, in lung cancer models as well. And so this slide then summarizes what we believe is going on, at least one hypothesis, that the treatment with an mTOR inhibitor of signaling downstream, in particular of cell lines that express the IGF-1R receptor, is that there's a feedback pathway that affects the signaling downstream of the IGF-1R receptor, that use of an IGF-1R inhibitor will block that feedback mechanism and have a more direct effect on signaling in these cell lines and then cell growth. So that's the preclinical data we had back in late 2008. And so a clinical trial was actually opened in late 2008, looking at the combination of the IGF-1R inhibitor with the mTOR inhibitor. Now, for those of you who don't know, phase 1 trials are the first trials where drugs are tested in patients, usually in highly refractory patients who don't respond to standard agents that are available to them. And so what you're looking for is obviously safety of these agents together, but also some preliminary activity. And so this combination was tested. We enriched for breast cancer patients based on the preclinical data we had, but we also wanted to ask whether other patients in other histologies might also be responsive to this combination. And we also were very good about examining biopsies from these patients to look at various biomarkers that I've already pointed out might be important in selecting patients who would be most likely to respond to this combination. So here's the results, data for which was just presented last Saturday at ASCO. And this is showing the data from a subset of the breast cancer patients. So overall, there were 23 breast cancer patients who were enrolled in this phase 1b study, and 10 of those 23 patients actually had some activity, anti-tumor activity, either partial responses based on RECIST criteria, or prolonged survival, or various tumor markers being knocked down very significantly. What was really exciting was the fact that not only do we see this sort of activity, which, by the way, typically in a study like this, you get excited if you see 10% response rates. When you see 10 out of 23, you start to get more excited. And what we found was that if you looked actually in those subset of patients that are ER positive and had the high proliferation index, that over 50% of those patients responded. So that certainly got our attention, and I think quite a few people's attention. And interestingly, if you look at the subset of patients that were ER positive but actually had a low proliferation index, 0 out of 5 of those patients had a response. This next slide shows the data from one of those patients treated with the combination. This is sort of pre-treatment. You see this very large tumor here with cross-sectional area here about a little over 4 centimeters. And you can see post-treatment here, 2 months later, the size of this tumor having shrunk significantly. As I pointed out, we also have data suggesting perhaps there are a subset of lung cancer patients who might also benefit from this combination. And here's data for one of those patients, a patient with non-small cell lung cancer who had previously failed 2 other lines of therapy. And you can see the very large tumor here that has shown over 50% reduction in the size of that tumor. And this patient remains on study a little more than 3 months after treatment. So this sort of study, I think, is important, not just for those people for us developing these agents, but others thinking about trying to try novel combinations of agents in the clinic. Because it shows that, clear activity can be seen with combinations of agents that when neither agent alone actually shows much activity. This is really getting at this idea of oncogene addiction, but perhaps that multiple pathways and hitting multiple nodes may be more effective than hitting any single node. A strategy that's been talked about a lot over the years, but this could be one of the first times that 2 agents that alone show no real activity as monotherapies, but together show really significant activity. And so discussions are ongoing with regulatory agencies who are still getting prepared to see this sort of data and try to understand how best to move it forward to help patients. I think what this does is also provide to senior management, to companies like ours, that such data can be very helpful in identifying subpopulations of patients that can be tested very early on. And what we've proposed is that it can provide this sort of very compelling biological and mechanistic hypotheses that can then be tested in vitro and in vivo. Given enough evidence, can then be tested very early on in clinical studies in sort of a paradigm-shifting way. And so one vision we have for the future about how best to do this is shown here. And it's really, as I've already said, based on the premise that tumors use multiple survival strategies. That's been talked about by Lou, by Scott, by others as well. And the idea is if you hit multiple nodes, you can apply maximal pressure to these tumors and really stop those tumors from growing. We believe that some of the new technologies, talked about the siRNA-based screening, talked about some of the high throughput technologies, and I'll talk about that a little more in just a second, are really important for identifying what are the right combinations of agents. So based on this sort of evidence, we've now gone forward and run enhancer screens, not just for mTOR and IGF-1R, but 5 other agents that are currently in our portfolio to try and find the optimal combinations of agents that can be used. We've also used it with other standard of care agents that are used in many different cancers. What we're also cognizant of is that there may not be agents for all the targets that we're looking for, and so this may also provide us with opportunities to find novel targets that could be combined with these agents to be able to be more effective in treating cancer patients. But the other thing we'd like to do, and using some of the new technologies, including robotics, that are available to us, is to screen for combinations of agents that may be more effective. And one way we'll be doing that is in a project we've called the Polypharmacology Project, which I'll just show briefly in this next slide. So the idea is to be able to look across a panel of different compounds, agents that are in clinical development, novel agents that we haven't taken forward yet, and see about what combination of agents would be most effective in treating cancer. So in the past, what we've had to do is just take this agent plus this agent and look at the combination. And we've done that for 20 or so different combinations. But with these high-throughput technologies now, what you can do is look across a much broader combination of agents. And so with the study we've just completed, I don't have results for today, but 53 different compounds we've now tested in 40 different cell lines, allowing over 1,000 different combinations of those various agents in 40 different cell lines, providing over 2.5 million different combinations together in those different cell lines. And this can be done with very few people and very quickly. So this screen was just run, a little less than 2 months. And again, hopefully invite me back to a future symposium and we can present more data on that. But what I can show you is the sort of data that we have already received from smaller sorts of screens like this, and how it's already having impact on subsequent trials that I don't have clinical data for today. But here's one example of combinations of agents that we've looked at, and in particular, we have an AKT inhibitor and a WEE1 inhibitor in the clinic today that we're testing. And what we've already noted is combinations with not just agents we have in our portfolio, but that other companies have in their portfolio. So for instance, we are combining our AKT inhibitor with a Merck inhibitor that AstraZeneca has generated based on the sort of combination data that we see across preclinical models. And this combination is now in the clinic. Data yesterday that was presented in a poster by Amy Gerton at Merck, presented on combinations with our WEE1 inhibitors, and data we're seeing preclinical, and these sorts of combinations are now being tested clinically. Again, based on the evidence that we've already presented on the combination of an IGF-1R plus mTOR inhibitor and the success that we're seeing now in the clinic, we have senior management buy-in to these to do these sorts of experiments in the clinic. So, in conclusion, I think it's really safe to say that the optimal benefits are going to be seen with combinations of therapies. The strategies that existed today were not optimal. Taking drug A plus drug B based on somebody's signaling diagram and trying to see whether they'll actually have some activity was probably less than optimal. But that we believe that optimizing these combinations using large-scale techniques and will allow us to provide better ideas of combinations of agents to be used in the clinic. So finally, I'd like to acknowledge the many people who have been involved in this study. There are a lot more than are shown here, but if I showed them all, they'd be in 4 font. You wouldn't read any of them. But I want to just highlight a number of people from Merck that are involved in this. Brian Hayes, ex-MIT, who leads the mTOR program. Sriram, who leads the IGF-1R program. Chris Winter, who leads the combination effort. Scott Ebbinghaus, who is a clinician who ran the phase 1b study. And the many clinicians on the phase 1 study from Val d'Hebron, Sarah Cannon, and the Cancer Institute of New Jersey. And most importantly, to the cancer patients and the very important role that they play in the development of agents such as this. And I thank you for your attention.